Now we have Tatek Dejene. He's going to speak about the survey of macrofungal diversity and analysis of edaphic factors influencing the fungal community of charch forest in dry Afro-montane areas in northern Ethiopia.
Okay, good afternoon. I'm Tatek from Ethiopia. In this presentation, I will speak on macrofungal diversity and the edaphic factors influencing the fungal community in the charch forest system of Ethiopia. So just to give you highlight about Ethiopia. Ethiopia is one of the African countries found in the eastern parts of Africa, and the country is characterized with a wide altitudinal variation and different topographic features. This altitudinal variation ranged from 110 below sea level, meters below sea level, up to 4,600 meters above sea level. So this wide topography, altitudinal variation and topographic differences, features provide the country to have a wide spectrum of habitat in which different flora and fauna can be found. And also this situation makes the country to be one of the biodiversity hotspot area in the world. So as to the vegetation aspects of the country, according to FRIS 2010, we have 12 different vegetation types, and these vegetations are classified based on the altitude in which they are occur. So we have those Afro-alpine vegetation in the very highland parts of the country, and up to the vegetation in the dry and arid and semi-arid parts of the country. In addition to this, we have also plantation forests dominated by exotic tree species like Eucalyptus, Cypressus, and pine species, both in the highland and the lowland parts of the country. So when we see specifically to the dry from mountain forests where we did our research, these forests are found in the highland parts of the country. When we say highland parts of the country, those areas which are situated at an altitudinal ranges between 2,000 and 3,500 meters above sea level. So from this picture, you can see that those areas marked with a yellow color are the highland parts of the country, and these areas constitute about 44% of the total land area of the country. And due to the suitability of this area for human habitation and agricultural purpose, majority of the populations are living in this area. The dominant vegetation type in this highland area of the country is called the dry Afro-mountain forests, and these forests are playing important role environmentally, because these forests are found in the mountainous area and provide important catchment protection. And also they are contributing for the socioeconomic of the local community, because they are the main sources of timber for the country, and also they provide different valuable non-timber forest products like mushroom, bamboo, and etc. However, due to this both ecological and economic significances that can be obtained from these forests, the dry from mountain forests are subjected to pressure in which their degradation is framed. This is more or less related with the growing population in the region, because as I have told you earlier, majority of the populations are living in this area, and there is a growing demand for forest products and the associated need for expansion of agricultural land for crop production, as well as for livestock rearing in the area. So not only the planing lands are suitable for agriculture are cleared, but also those mountainous areas where the dry from mountain forests are found has been converted into agriculture crop production purposes. Because of that, currently the dry from mountain forests are restricted to inaccessible areas, and also they are found only around the churches. These are the churches, and these forests are called church forests, remnant dry from mountain forests. Generally, we call them church forests. So these church forests are conserved because of the spiritual or religious value associated with them. This is because these forests serve as a monastery and also a graveyard for those Christian believers. So we wanted to study macrofungal communities of these forests, because different research activities have been done in order to assist the rehabilitations of this type of forest system in the northern parts of the country. But most of these researches are focused on the above story components of this forest system, mainly focused on the three components. But nothing has been so far done regarding the understory and the below ground components of this forest system, like a mushroom, which have both ecological and economic significances if you manage and conserve these species. So we wanted to study the macrofungal species in these forests because in order to enable the integrations of macrofungal species into the wider conservation strategies currently launched to conserve and rehabilitate these forests. And also we wanted to know what action must be taken to conserve these macrofungal species in this forest system. We had three objectives. The first one is to describe fungal species richness, diversity, and sporocarpus production of this forest system, to correlate the macrofungal and plant diversity of the church forests, and also to determine which edaphic variables are more responsible for governing the macrofungal taxa composition in these church forests. We did these experiments in three church forests, which are located in the northern parts, namely Taragadam and Alamsaga forests. And in these church forests, we establish about 27 permanent plot with the area of 100 meters square, and 9 plot in each forest. So after establishing the plots, we collected the whole sporocarpus that we found from each forest for tax identification purpose, as well as for fresh biomass measurement. And in addition to this, we also took a composite soil sample from each plot that we established in the forest. And finally, we did a biodiversity analysis, and we did an ordination analysis, and also we did a vegetation analysis. As to the main findings from this result, We collected a total of 250 macrofungal taxa from the entire forest systems. And these taxa are dominated by saprophytic species. And of the total taxa that we collected, 8% were ectomycorrhizal. And of the total taxa, we have got about 68 species were found to be edible. These are some of the taxa that we collected, like termitomycetes, edible species, Tricoloma, agaricus, different types of agaricus species that can be used as a food. As to the taxa richness and the diversity differences, we have got both the diversity and richness difference among the three forests that we compared. And also we try to correlate the taxa, the species, the vascular plant richness and the diversity indices of the vascular plants with the macrofungal species, and we found that the species, vascular plant species richness is directly correlated with the diversity of macrofungal species in the study. And also we assessed the sporocarpus production in each forest, and we found some kind of differences, as you can see from the pictures. And also from the taxa composition, we found the organic matter, CEC, and the pH are the factor, the edaphic factor that govern the taxa composition in these church forests. So as a conclusion, the diversity indices and community composition of macrofungal were influenced by site condition, including the vascular plant diversity and soil fertility gradients of the church forests. Thus, the promotion of vascular tree diversity in church forest system by enrichment planting or natural assisted enrichment systems would offer suitable habitat with a variable microclimate that should assist the macrofungal species diversity and production in the area. And also our survey also revealed that the presence of valuable macrofungal species belongs to the Tricoloma, Cius, and the Termitomycetes genera, which could potentially be marketed and hence could provide supplementary income to forest-dependent local people in the area. So I want to acknowledge ACID for the financial support for this project. And also the Ethiopian Environment and Forestry Research Institute for logistics support. And finally, thank you for your attention.